Hello everyone, I am Ida Höijer and I'm a PhD candidate from SciLife Lab, Uppsala University. Today I will talk to you about how we are using single molecule sequencing to study Cas9 induced off-target effects in vitro. The CRISPR-Cas9 system quickly became an immensely popular tool for genome editing after it was first discovered. It is now widely used in both functional genomics, bioengineering, medicine, and so forth. The CRISPR-Cas9 complex induces a double-stranded cut in the DNA by the Cas9 enzyme that is guided by target-specific oligo, or the guide RNA. It is a concern, however, that Cas9 can induce so-called off-target effects caused by unspecific binding of the guide RNA. There are in silico tools to predict these off-target effects, but they are typically based on sequence comparison. This is a quick and easy approach, although it is difficult to predict guide RNA binding. You will also need a reference genome, and to this I want to add that this reference might differ from the actual genome that you are studying. In this slide, I want to address guide RNA binding and genome variability. You might have this guide RNA that binds perfectly to your reference genome, but the individual you are studying, there might be a bunch of SNPs, shown by the red arrows here. So the same guide RNA might not bind anymore to your individual. However, it might bind in another location in this genome. So the question here is, can we really predict off-target effects if we don't know the exact sequence of the genome that we are studying? To deal with this issue, we developed a protocol using single molecule sequencing that detects guide RNA off-target binding that we call off-target sequencing, or OTS for short. So the PacBio version of this protocol we are calling SMRT OTS. And now I'll just quickly go through how this protocol works. So what you first do is more or less a conventional SMRTbell library with random sharing and ligation of sequencing adapters. So some of these molecules will contain your on-target, and some of them will contain your off-targets, which is shown in red and green here. In the next step, you induce the Cas9 digestion, and in this step you can use either just one guide RNA, or you can pool a bunch of different guide RNAs that you want to study. Then you ligate a different kind of adapters called capture adapters onto these broken molecules that were cleaved by Cas9. And in the next step, we have magnetic beads that can pull out all the molecules with the capture adapters, and this is how we enrich for these molecules. And then we can go ahead and sequence. I just want to add that we also have a version of this protocol for nanopore sequencing that we called nano-OTS. The reads you get when you're using this protocol have really specific read pattern, as you can see here. Basically, all reads start or end at the same position where the Cas9 site is. And this is true for both on and off targets. This is also something we use in our pipeline for the analyses of these reads. But the first thing we do after sequencing is mapping all the reads to reference. And then we make this software that we call Insider that is able to identify all these specific read patterns. So we're able to detect all of our on-target sites, but more importantly, the off-target sites. So as I mentioned in the previous slide, you can use either one guide RNA or several guide RNAs when you do this assay. But if you use several guide RNAs, it's important to assign the right guide RNA to the right OTS site. So what we do is that we obtain the reference sequence for all the OTS sites, and then we do a pairwise alignment of the guide RNAs to be able to distinguish what guide RNA has caused the specific OTS site. And then we can compile this into a table format and visualize this. So the genome we studied in our project is from the HEC 293 cell line, which is an embryonic cell line that was first cultured in the 70s. To get a comprehensive map of this genome, we perform whole genome sequencing using HiFi reads. In this way, we are able to take SNPs and larger structural variants in the genome. So using two SQL2 cells, we got 18x coverage of this genome. So now we are able to get the exact genome sequence for where our guide RNAs are binding. The experimental setup for our project is that we performed SMRT OTS and non-OTS for our cell line with 3 different guide RNAs, the ATXN10, the MMP14, and the NAK1. These are guide RNAs that we have been using or are currently using in other projects. So after running our OTS protocols, we ended up with 55 guide RNA binding sites for all of our 3 guide RNAs that were detected with both technologies. Most of the sites were for ATXN10, but also a bunch for MMP14 and only a couple for NAK1. Something that is interesting is that we identified off-target sites that had up to 5 base pair mismatches between the genomic sequence and the guide RNA. If we look more into the specifics, we can find evidence of allele-specific guide RNA binding. On the bottom here, we have the HiFi data for a cell line. You can see 2 different haplotypes. We have the reference haplotype that had a lot of SNPs, but we also have an alternative haplotype that only have 3 SNPs. And if we look at our OTS data, on the top, we can see that we only have reads from the alternative allele. This means that the guide RNA does not bind to the reference haplotype, which has 3 mismatches between the guide RNA and the genomic sequence. However, there's only 2 mismatches between the alternative allele and our guide RNA, and in this case, we have binding. So the conclusion is that genetic variation can induce guide RNA binding, at least what we see here in vitro, which means that different individuals can get different type of target effects. What we also did in collaboration with Jason Shen was to assemble our HiFi reads into a reference genome for our HEC 293 cell line using his new assembly tool, Peregrine. By doing this, we were able to get a complete de novo genome, and this of quite high quality for this cell line. We then mapped our off-target sequencing data to this genome, and we were able to recover more than 98% of our off-target binding sites. This means that we can also use this method in organisms where we don't have a known reference genome. And to conclude this presentation, with our off-target sequencing data and our HiFi data, we are able to generate a detailed map of Cas9 cleavage sites for our guide RNAs and cell line. We're also able to detect allele-specific guide RNA binding. We can also do this in organisms where we don't have a reference genome. This said, we will still need to learn if this also represents what is happening in living cells and in organisms, and this is something that we are actually currently investigating. If you want to learn more about this project, we have a preprint out on bioRxiv, and please go ahead and check that out if you're interested. So with this, I want to thank everybody involved in this project, especially my co-supervisor Adam Mamar that have put a lot of work into this project. Also thanks to Jason and some people at PacBio for supporting us along the way. And of course, I would like to thank you for listening. Thank you.